previous lecture we have discussed about ion exchange chromatography. In ion exchange chromatography, the property of charge which a particular analyte carries on itself is utilized for separation procedure in chromatography. In this lecture we are going to discuss another important chromatographic method, that is gel permeation chromatography. Now, gel permeation chromatography utilizes the molecular size or shape of a molecule for separation. This is also called, this has different names, and this is also called gel filtration chromatography or gel exclusion, or we can say size exclusion chromatography, then molecular sieve chromatography, or even simply gel chromatography. Now, this technique is a form of partition chromatography, and it is used for separations of molecules, like I said, on the basis of their shape and size. Now, this technique exploits the molecular sieve properties of a variety of porous materials. If we consider basic principle of these techniques, then different kinds of molecules which differ in sizes are partitioned between solvent and stationary phase of a defined porosity. Now, the separation process is carried out using a porous gel material or gel matrix in bead form, and it is packed in a column surrounded by the mobile phase or the solvent. Now, if a sample is applied which contain mixtures of smaller, larger and intermediate size molecules compared to the pores of the stationary phase matrix, then separation will occur as one where smaller molecules can enter the pores and hence move slowly through the column. This means that they will spend more time on the column, because they can move in and out of the beads more frequently than other molecules. So, the smaller molecule will rather spend more time on the column as compared to other molecules. Now, larger molecules which are too large to enter into the beads due to the pore size limit, they will be completely excluded from the stationary phase and hence elute first from the column, and they will come what we call in void volume. Then there are molecules which are intermediate in size and can enter the stationary phase, but spends less time. You can say that they spend relatively less time than smallest molecules and more time than the largest molecules. So, depending on their molecular size, there will be a kind of relative time spent in the gel or in the column, and accordingly they will be purified. So, all the molecules which are eluted, they are eluted in order of their decreasing size. Now, let me explain this before we go into the details of this method. Let me explain in a very brief this method to you on your screen. All right. So, now first thing is that this chromatography technique is based on size and shape, and then another thing is, since there is molecular size is the basis of separation, the column lengths are usually long. So, that the analytes needs to be separated, can be separated well, as they will travel a longer distance, that will allow them to be separated to a certain extent that peaks do not overlap. Second thing is that the column is mostly narrow bore column, or it has a lower what you call diameter of the column is less as compared to other normal columns. So, that there is no diffusion and gel filtration is run successfully. So, what you have is, now the beads which are packed, that is the most important part here, that is beads if we say. So, if there is a column, and like I said, it will be a long column and with narrow bore size or diameter, and this column will contain beads here. Now, what is the property of the bead, like what is the basis of separation here, like we were talking about. So, now these beads are porous beads, these are hollow beads actually, and these beads, if I make it here a single bead, then these beads are totally hollow inside and they have a particular pore actually. Pore means, this is just a schematic to tell you, these are cross linked polymers and they present a gel like structure and they have a particular porosity because of this cross linking. And porosity, just like to make you understand, if this is the pore size, then what will happen? A molecule which is bigger than this pore size, if I say this is the molecule, then this cannot enter here, but a molecule which is smaller than pore size can enter into the pores. This can go into it can access the inner volume of this bead, which is the trapped solvent or trapped mobile phase, but the larger molecule cannot enter. So, what happens then? That larger molecules will not be able to spend time, it will only spend time in interstitial spaces or the space which is surrounding this stationary support material, which is in which the stationary phase is contained. So, this will come very fast, but the smaller molecule will spend more time because it will go back and forth as it travels down the column, it will move back and forth into the stationary phase and then mobile phase. So, if you compare different sizes of molecules, if I say there is one size, there is another size, there is another size, there is a larger size, then all of these molecules according to their molecular mass or molecular weight, they will spend relatively different times, smaller the longest time, the largest the least time, and in between they will be spending different times as per their size. So, that is how the separation occurs, because they are moving down, though you have a continuous flow system and they are moving down at different speeds or at different rates actually, which are eluted at different rates. So, this is a very simple basic principle of column chromatography, of gel filtration chromatography. Now, these columns, like I said, are long and they are narrow bore. So, they could be for low pressure, there could be coarse beads or bigger beads, and for high performance liquid chromatography, there could be finer beads which are packed very nicely. Now, this system mostly cannot be run by gravity feed. So, it is you require here, if I have certain things, you require a pump here, which is a very important part of this column, so that it can give you a constant flow rate. So, this is a prerequisite that you should have a pumping system to run the column. If you use certain like over you can put a reservoir sort of over the column, but that will not give you a constant flow rate. So, this is a very in a simple method I tried to explain the principle of this gel filtration. Now, let us get into the our discussion or detailed discussion. All right. So, what we were talking about was that smaller molecules can enter the pores, hence they spend more time in the column, and larger molecules are excluded. So, larger molecules are excluded in void volume or dead volume, which is the total volume of the column minus the volume occupied by the beads. So, since it is not getting inside the beads, it will be excluded. Now, here there is another figure which explains it the same principle. If you can see here, this is a column where these are beads actually, and you have different size molecules. There is a larger size, intermediate size and a smaller size. Now, if you can see them, they are moving at different rates, larger moving out very fast, intermediate moving at a relatively slower speed than the larger ones, and the smallest one will move at the slowest speed. And as the column runs and as the time passes, the first to elute is the larger one and you get a separate fraction. That is how you can see very clearly. The next one, you can see how the peak resolution can take place if their molecular sizes are farther apart, far apart, and they could be easily separated into different fractions and could be utilized for different applications as a purified product. Now, what I was trying to say, why you need a long column? You need a long column as they start a single entity in the, when you load these columns, and as they move, if you have a smaller column, then their separation, like here it is not really separated quite a lot, the separation will be not complete, and they will have, you can have say overlapping peaks. So, as long as the column is, and narrow bore column for avoid diffusions, you will get the better separations could be achieved in gel filtration chromatography. So, this is very, in a very simple way, this explains the whole basis. Now, if I have to say like what are different terms in columns, if you see here the shaded part here, this is called the void volume. It is the volume outside the support matrix. This is total volume, like it is taken both the volume occupied outside the beads and inside the beads, that is the total bed volume. And then you have volume which is inside the stationary phase, and that is called Vs, or volume of the solvent held in the beads. So, that is the kind of distribution, or you can say that is how the void volume, the total bed volume, and the volume occupied by solvent inside the stationary inside the support beads is described. Now, here elution volume of a large totally excluded molecule will be given by void volume. Vt is physical volume, and other, like I said, is the you can divide, or you can subtract the total bed volume by the void volume, and you can get the volume of the beads. Now, elution volume, there is another term called elution volume, and that volume is the volume between the point of injection of the sample analyte at the top of the column and the peak maximum, which is dependent on the fraction of stationary phase available to the analyte for diffusion. So, that will be like elution volume is the volume which is required for eluting the particular analyte, and that will be starting from the point where you inject the sample and the point where a peak appears, or that analyte is eluted. So, if you consider here for these equations here, Kd is of course, it is a distribution coefficient, and Kab is proportion of pores available to the molecule, and both could be used. So, for a totally excluded molecule, since Kd or Kab will be 0, and so the elution volume will equal the void volume, that is for completely excluded molecule. Totally included molecule will be, it is like there is no fractionation happening, and elution volume will be almost equal to the total volume of the column. So, and the intermediates could then be separated in between, they will have certain values in between 0 and 1. So, a total bed volume can be measured by determining the elution volume of a small molecule, that is which is not in the fractionation range, like salts actually or acetone. They could be utilized since they have complete access, they can easily move in. So, they just move with the mobile phase, and they spend the most time, and that could be calculated, elution volume could be calculated by utilizing a very small molecule. If you consider the efficiency of a column or a gel filtration column, if you could recall, we were talking about the theoretical plates actually. Theoretical plates, more is the number of theoretical plates, better is the resolution, better is the separation power of a column. So, here efficiency is determined by calculating number of theoretical plates per meter, and you can this equation could be utilized for calculating the theoretical plates. where V is the elution volume, Vw is the peak volume at half the height of the peak, and L is the column length. So, it is like here half the height of the peak and this is the elution volume. So, you can calculate the theoretical plates. Remember, if you compare low pressure and high performance liquid chromatography, though the length of the high performance liquid chromatography columns are not that long, but still they contain more number of theoretical plates, because the material which is packed, the stationary phase or support matrix is much finer material, and so they can pack in many or more material in a small area, and thereby providing more number of theoretical plates and enhancing the resolution or the separation power of the column. Now, let us discuss all different aspects of the gel filtration chromatography. First is gel permeation media. Now, when you take the media, there should be certain properties of this media, that is column material. One is that this should be completely inert with respect to the molecules being separated. If it is not inert, then non-specific binding or adsorption might be a big problem. So, the media or the column material should be completely inert for better separation. There are a lot of different kinds of media or gel material which are utilized. If you could recall, we have discussed about this, but as we will go along here in terms of gel filtration, there could be lot of different support media or gels are available. Dextran is a homopolysaccharide of glucose residues, and it is prepared with various degrees of cross linking to control pore size. Remember, here it is a sieving effect which is or filtration effect, it kind of filtration which is taking place. So, pore size is very important, as on particular pore size of the gel material, the different kinds of molecules will be separated. It is like cross linking is controlled for pore size. It could be obtained as dry beads or beads swelled when water and it could be the beads could be swelled when water is added. We will see how that is done. Now, this trade name is Cefadex, mainly used for separation of small peptides and globular proteins with a small to average molecular mass. Now, when we say we have to swell it, the swelling is done like you take the dry beads which are supplied by commercially. Now, those could be put in the water or buffer initially in the water and left for certain period of time, say you can leave them overnight or so and let them swell. The swelling could be observed by, you can look into the under the microscope and you will observe the unswelled and swelled beads could be clearly observed. It could be enhanced by, you can put this beads in water in an oven at higher temperature, they will swell faster. So, that could also be done. Then there is polyacrylamide. These gels are prepared by again cross linking with the acrylamide with N,N-methylene bisacrylamide. The pore size is determined by the degree of cross linking and the separation properties of this material are mainly same as that of others like dextrans, and they are supplied as, for example, Biogel P. They are available in wide range of pore size. So, you can isolate or purify different range of material or analytes. Then there is agarose. It is a linear polymer of D-galactose and 3,6 anhydro-1-galactose. It forms a gel that is held together by mostly hydrogen bonds, and the concentration of the material in the gel determines the pore size. So, the pores of the agarose gel are much larger than those of Cefadex or Biogel P, and it could be utilized for separation of large globular proteins or long linear molecules such as DNA. Dextran and agarose beads are biodegradable. So, many times you have to use antimicrobial agents like sodium azide for storage. Like another important part as we were discussing was large globular proteins and long and linear molecules, like I said. This will also affect, like shape of the molecule will also affect the separation. Remember, when we consider all these things, we ideally take spherical shape, but maybe two molecules of the same molecular mass, but differing in shape, like say a molecule which is spherical and a molecule which is elongated. Elongated molecule will certainly will not be able to enter the beads as compared to the same molecular mass spherical material. So, those things has to be taken into account while purification. So, all when you are standardizing the things, mostly it is done for spherical molecule. So, when you are considering gel matrix, like different materials we have discussed, there are certain factors which needs to be considered while you are choosing a gel matrix. One is fractionation range, that is, if all proteins in a mixtures are to be separated from relatively low molecular weight solutes, like say your proteins are large molecules and you have to separate a smaller molecule from the mixture, you have to separate your protein from the smaller molecules, then you can have small packings with small pores or gel material, and this is kind of a process called desalting, where proteins are totally excluded because you are using the gel material which is of very small pore size. So, your material does not, or your protein does not enter, but the smaller molecules will enter and you will be able to easily purify this. Like for example, Cefadex G25 or Biogel P6 or P10 could be utilized for this purpose. For resolution of different proteins which are more closely related in size, there is a range of size actually, then you have to take a matrix where it is like molecules do not get excluded in void volume, or they are not elute in void volume, and so there is a fractionation range of the gel, that is of a particular pore size, and it allows all the analytes to enter unless you want specifically that one of them is excluded from the excluded and does not fractionate actually. Then you have to consider resolution. For higher resolution, smaller beads should be utilized, they will give better resolution because molecular diffusion is slow, is lower, and there is less zone broadening. However, smaller particles gives higher resistance. So, you have to use say high performance chromatography or very good pumps for that purpose. Rigid and larger beads allow faster flow rates, and many times they are used for large scale applications. So, this is another factor that what kind of resolution you are seeking for, and you can use either low pressure chromatography or high performance chromatography. Now, another factor, very important factor, is stability of the matrix. Matrix needs or should be stable at different pH values or in different buffer and organic solvent conditions, because you are going to use different kinds of buffers or solvents during the run, and the matrix needs to be physically and chemically stable for these solvents, and necessarily proteins or any analyte which needs to be purified should not be affected by this particular problem. So, this is another important part, that is stability. Now, if you consider the instrumentation of gel filtration, it is a very simple or very basic sort of thing, as we have already discussed earlier in for low pressure chromatography and again for high performance liquid chromatography. In instrumentation part, the basic part is you require a column which is like I said, it is a long column and narrow bore column. You require a detector so that you can go through the peaks which are coming and you can see the purification visually. Then you require a fraction collector to collect those fractions or different peaks separately. Then there is flow rate has to be controlled, and it is mostly like I done. You can have a constant head reservoir, but that is not very suitable. What you require a pumping system, it could be for low pressure, it could be peristaltic pump, or for high pressure it could be like constant displacement or other pumps could be utilized. Now, the system should be arranged with the monitor connected as close as possible to the column outlet, and this is to prevent the mixing or longitudinal mixing in the tubing, and similarly the connecting tubings and flow cells should have narrow dimensions to prevent mixing. So, that is a very important part. The column needs to be like when you are purifying this, the aim of the gel filtration is to obtain greatest possible resolution of the molecules of interest, and the resolution will be improved by the distance between the separated zones and their zone divided by zone width. So, what you have is like I said earlier, longer the column better is the resolution, because you give more time for the analytes to separate. So, that when they travel through the mobile and stationary phase, the smaller columns, they will the peaks will overlap, but as the column length goes up, then they will have more time to separate from each other and separate peaks could be obtained. Then columns, if you say longer columns, it up to 100 centimeter length columns could be utilized. The internal diameter, like I said, it is very small, like 1 or 1.25 centimeter or so, and mostly what is done is it is taken, if you take then the length should be 20 to 40 times longer than the wider. So, that has to be remembered, that you have to have a longer columns with smaller diameter, internal diameter of the column. When you are preparing the gels for column chromatography, what is commercially provided is either you will get dry powders like say Cephalodex or others, and or you can get the swelled or in certain medium, liquid medium, that particular matrix. If you are provided with dry powders, then dry powders has to be kept in a beaker or container. In the it has to be swelled in the water, and like I said, you can swell it overnight or you can swell in oven, which could be faster, and you can check for whether beads are swelled uniformly by observing them under microscope. Gels which are supplied pre-swollen like Cefacryl and others, what you have to do is you require is that you have to exchange them in the buffer, which is the proper eluent buffer before use, and then when you are again storing them, again you have to store them in the in particular solvent, say ethanol or others, for long term storage. So, this is very important. Now, packing is another important part of running a column, and here packing of the gel filtration column is not that easy if you compare it to the ion exchange column. Ion exchange columns are smaller columns, and you do not have to worry about length and width and the diameter so much, but here since these are long columns and with a narrow bore or a smaller internal diameter, you have to be very carefully pack the column, so that column is packed uniformly without air bubbles, without any gaps or like say certain cracks, so that run could be proper. So, gels should consist of a slurry when you are packing this of approximately 75% settled gel and around 25% supernatant liquid. Now, suspensions will be at constant done at constant operating temperatures, and like I said earlier, if you have different buffers can change their pH, and even there could be certain other properties changed due to temperature. So, that has to be taken care. Like for example, if you suddenly take it into the cold room conditions, certain things could swell or or could shrink, those could be another problems. So, suspensions should be at constant operating temperature before and during packing, that is a very important. So, if you want to do the experiment in cold room, then you can swell the gel in the cold room, swell the gel first and put it in the cold room and then pack it. Now, air bubble formation and other things should be prevented during column packing, and which can be monitored by using color test sample such as like for example, blue dextran. Blue dextran could also be utilized for determining the void volume, as it will not enter the beads and it is completely excluded from most of the gel matrix. Now, the packing is mostly done with a pump, like so you have all the material is in a particular container over the column, it is packed inside in Marriott flask, and it could be connected to a pump and it could be packed properly. Manual packing might not be a proper or good idea. So, it has to be packed with a peristaltic pump or other pump at a constant flow rate, and it is packed properly. Remember, low pressure beads, if you put too much of high pressure, they will flatten and then no separation will take place. So, that has to be taken care that they are packed properly, they are not subjected to very high pressure, which could be which will be like totally damaging them, and no proper purification will take place. Then choice of eluent for running the column is important, and when you are choosing an eluent, it should be one which can where your analyte could be stable, and you can add various cofactors or metals or other things without any problem, without any interference. So, that is very important. If the sample is to be freeze dried, then you can use volatile buffers. Like say you want to lyophilize the sample after the purification, then if you directly use the volatile buffers, you really do not have to exchange the buffer later on. Like say ammonium bicarbonate could be utilized, or water which is volatile, or other buffers could be utilized. So, that is a very important part if you need to lyophilize. Now, one of the most important part of this gel chromatography is the sample size, or how much amount needs to be loaded onto the column. Now, the sample volume should be as small as possible and must be lower than the separation volume between components in order to get higher resolution. Typically, it should be 1 to 5% of the total wet volume. So, larger volume will not give you a good separation, I will tell you why. And so, you have to load the sample into in a higher concentration, but maybe what you call smaller volume actually. For group separation, where you are not so much worried about separating many analytes, you can maybe use little larger volume. So, when you sample, when you have is, so gel filtration like is independent of sample mass, and so high concentration could be utilized here without any problem, and you can get high resolution. However, the concentration which can be used is generally limited by the viscosity. If it is a too viscous a sample, then there could be a problem relative to the eluent. So, high viscosity will result in zone stability and irregular flow pattern. So, the protein concentration of the sample applied to the column could be around 10 to 20 milligram per ml. Now, let me first tell you why the sample volume is very important in here as compared to other chromatographic techniques, where larger volume could be utilized in the column. So, now let us understand this. Supposing you have a column here, and this is your beads here in the column. I am just trying, this is just a schematic, this needs to be longer column. Now, this one you have your sample, say if I have a larger volume of sample, why I am saying why you require a smaller volume, and you have a different size things here, all right. Now, what happens is that when you have a larger volume of column, this bead here which is entering in the column, or this large size analyte which is entering the column, and this analyte here is entering at the same time, all right. So, they will be separated accordingly, but what happens is these, when they have entered here in this beads, this will pass through interstitial space, this will go through this into the bead and spend certain time, but there will be a time gap between these two entering and these two entering actually. So, what will happen, as this one has travelled, but it will travel farther, but this one will, since this like addition to the void volume, if suppose this is excluded, then this is addition to the void volume, and so there could be a problem when you have many analytes of peak overlaps or peak broadening, because peak broadening will you will not get a sharp peak or this sort of peak, rather you will get a spread peak. And what is the problem is, because these will be entering at different times. So, the purification will not be proper. So, it is very important that sample is applied in a very small volume, so that all of them, all analytes almost enter the gel at the same time, and then they separate according to their retention capacity or retention times, actually retention volumes, as per their molecular size. So, that is a very important factor actually which needs to be looked into. So, this is another part which is important. Now, when you are applying sample application here, sample application as we have discussed in ion exchange chromatography, here also you have to take a lot of care when you are applying the sample. So, care should be taken when applying the sample to the surface of the bed, and one is that bed should not be disturbed. If you are disturbing the bed, then again you have to wait so that it settles down, and it might settle down not so uniformly. Now, when samples needs to be applied, first is you can remove the or drain the bed surface on the top without disturbing the bed surface, and then slowly very carefully put your sample on top of the bed. Then you can layer the sample under the eluent if it is viscous, little bit, like say high concentration proteins will not, unless you mix very harshly, they will not mix, and evenly it could be layered in the eluent, like I have told you in ion exchange chromatography technique also. Or you can use the loop injector or three-way valve could be utilized for this purpose, as we have discussed in high performance liquid chromatography. So, there could be different ways. So, if you are applying it like manually, then the above plunger needs to be again mobile phase needs to be put in after the sample has entered the bed or the stationary support material, and then you can layer the mobile phase and close the column and connect it to the pump. So, the maximum resolution is obtained like I said, using long columns, using slow flow rates, and using concentrated but less volume of the samples. Optimum flow rate could be like 2 ml per centimeter square per hour, but it could go very high for say cefacryl like 30 ml per centimeter square per hour. But remember, for low pressure chromatography, if you use higher flow rates, then beads could be flattened because of much higher pressure. So, those things has to be taken care of. Once you have utilized the column, your analytes are done, you can re-equilibrate the column for next run, or otherwise gels may be cleaned up. You can treat it with say 0.2 molar sodium hydroxide or certain other non-ionic detergents, clean it up, and then you can store it in the presence of antimicrobial agents like sodium azide, which could be put in a very small amount, or say 20% ethanol can also be utilized. So, this way you can easily store your material. Now, this was the basis and you can say mechanism and working of the gel filtration chromatography. Gel filtration chromatography has lot of advantages or lot of application. Now, before like so, when you are utilizing this method, it is the best method for separating molecules which are differing in molecular weight or molecular size. So, here like temperature, pH, ionic strength, buffer composition, etc., will not be making a difference. Like in ion exchange, you have to have a particular buffer condition or pH condition, because they will be charged accordingly. Here only you have to worry about whether your analyte or protein is stable. So, that is the important part. And separation can be carried out under any conditions, mostly it is isocratic flow, like it could be in the presence of ions or cofactors, detergents, and other particular conditions could be utilized. There is a very little adsorption, because these are inert columns material, it is a lot less zonal spreading than other techniques, elution volume is related to the molecular weight. Like I said, so this could be utilized for say determining molecular mass. If you consider the applications of gel filtration chromatography, there are many applications of gel filtration chromatography, and let me list you some of them. One is, like we were discussing, purification. Purification is the most important application of gel filtration chromatography. Now, it is like main application, and you can separate them on the basis of their shape or size. Proteins, enzymes, hormones, antibodies, nucleic acids, polysaccharides, and many more could be separated on or purified on this chromatographic column. In general, gel filtration is considered a low resolution chromatography, and it is not the first step in the chromatographic procedure when you are purifying an analyte, rather it is the final step. But in certain cases, it depends from case to case, you can use it before also, but mostly it is the final polishing steps actually. Relative molecular mass determination could be performed on gel filtration column. So, what happens is, like I said, elution volumes of globular proteins are a characteristic value, and they could be determined by the relative molecular mass. So, if you run standards, certain standard proteins which you know the molecular mass and you know the elution volumes, then you have a standard graph which could be plotted, and then an unknown sample could be run, and through the calibration curve the proteins of similar shape or like spherical proteins ideally, their molecular weight could be determined. So, that is a very good technique to determine the molecular mass. Only thing is that all the standard conditions which have been maintained for running the standards needs to be maintained for running the unknown sample. So, this is how you can plot log of molecular weight and the elution volume. There will be a plot, and from this plot you can determine the unknown sample. You can concentrate certain sample here, say you have a purified sample or other. So, what is done is, solution of say high molecular weight substances can be concentrated by simply adding dry Cefadex G25. Now, as earlier I told you, these are not for purification, but kind of desalting. So, what will happen when you put the dry powder, it will swell and absorb lot of water, and so low molecular substances will be in the gel, but high molecular substances will be excluded and they will remain in solution. So, after certain period of time, you can at a very low speed you can centrifuge them, and you can get the solution or the solvent which contains your high molecular weight analyte, and it is concentrated because a lot of water has been absorbed by the material. This could be utilized, it is frequently utilized for desalting. So, by use of Cefadex G25 solutions, high molecular mass compounds may be desalted, where you can pre-swell the gel in this case. So, the high molecular mass substance will just move in the void volume, whereas the salts will be distributed and will be in the stationary phase. So, this could be another important part. It is very fast and more efficient method than dialysis, which is a little cumbersome. There are a lot of applications of this particular method. You can remove, like for example, removal of phenol from nucleic acid preparations, it could be utilized. It could be utilized for removal of ammonium sulphate from protein preparations. It could be utilized for removal of salts from samples eluted from ion exchange. Like for example, ion exchange will contain a lot of salt, and to take it to the next level you need to remove the salt in certain cases. You can do protein binding studies in here. Protein binding studies, like you can be use the gel filtration chromatography to study the reversible binding of a ligand to its to a macromolecule. And there are a lot of combinations, like say receptor, hormone, or say protease or inhibitor, Likewise, it could be utilized antibody antigen, and these could be like when you will have a separate ligand and protein, they will have a different elution volumes, but when they combine and if they are interacting with each other, then elution volume will be different, because they will be eluting earlier as per increase in the molecular mass. So, there is a procedure and you can like go through that, and even experiment could be repeated as a series of ligand concentration, and you can appropriate binding constants can also be calculated. So, it is a very useful technique. You can also determine quaternary and tertiary structure here. So, what is done when you have purified proteins, and if you run through the gel filtration column through the elution volume or through the retention time, their hydrodynamic radius of typical protein will play a role, and so there will be change in elution volumes, and so two forms could be separated, as well as you will have an indication there is certain change in the quaternary or tertiary structure of a particular protein. So, that is another way. Many times in polymer synthesis it could be utilized to measure the size and the polydispersity of a synthesized polymer. So, that is the ability to be able to find the distribution of the size of the polymer molecule, and if standards of a known size are run previously, then a calibration curve can be created to determine the size of polymer molecules of interest in the solvent. So, these are different applications of gel filtration chromatography technique. So, as you see here in this is a very useful technique, and it is has many applications. It is on the basis of size or shape, like I said. Ideally we talk about spherical size, but if it is elongated shape, then certainly same molecular mass protein will be excluded faster than a spherical protein. So, this is a very simple, very nice technique, could be done in low pressure chromatography or high performance liquid chromatography with different types of columns with more number of theoretical plates as their finer particles are packed. And it is like one of the techniques where you do not have to worry about certain factors, physical properties except for size here, like pH or other things. Only thing to worry about is stability of the protein in particular buffer condition. So, this completes the gel filtration chromatography. In the next lecture, we are going to discuss about another important technique which is affinity chromatography. Thank you.